फोटोसिन्थेटिक पाथवेज यसमा चाहिँ हाम्रो C3 र C4 साइकल पर्छ. डार्क रिएक्सन or Blackman's reaction or path of carbon in photosynthesis. The chemical process of photosynthesis occurring independent of light. अनि यसलाई चाहिँ डार्क रिएक्सन भनिन्छ. It takes place in the stroma of chloroplast. In dark reaction, the sugars are synthesized from CO2. The energy for CO2 is fixed to energy-rich carbohydrates using the energy-rich compound ATP and the assimilatory power NADPH2 or light reaction. अनि हामीले चाहिँ यसलाई कार्बन फिक्सेसन अथवा कार्बन एसिमिलेसन भनिन्छ. In dark reaction, two types of cyclic reaction occurs. Calvin cycle or C3 cycle अनि H and slag pathway or C4 cycle.
This is a complete diagram of C3 cycle. In C3 cycle, RUBP ribose 1,5-diphosphate combine with 1 CO2 to form 3 phosphoglyceric acid in presence of carboxy dismutase. Again, this 3 phosphoglyceric acid forms 1,3-diphosphoglyceric acid in presence of triose phosphate kinase, and it also convert 1 ATP into 1 ADP. This 1,3-diphosphoglyceric acid again convert into 3-phosphoglyceraldehyde in presence of triose phosphate dehydrogenase. Again, this 3-phosphoglyceraldehyde convert into fructose 1,6-biphosphate in presence of aldolase. This fructose 1,6-biphosphate forms fructose 6-phosphate in presence of phosphatase. Again, in presence of transketolase and 3-phosphoglyceraldehyde, fructose 6-phosphate forms erythrose 4-phosphate and xylulose 5-phosphate. Again, in presence of aldolase, erythrose 4-phosphate convert into pseudoheptulose 1,7-diphosphate. This pseudoheptulose 1,7-diphosphate convert into pseudoheptulose 7-phosphate in presence of phosphatase. In presence of 3-phosphoglyceraldehyde and transketolase, pseudoheptulose 7-phosphate convert into xylulose 5-phosphate and ribose 5-phosphate. In presence of phosphopentokinase, ribose 5-phosphate convert into ribose 1,5-diphosphate, and again ribose 5-phosphate convert into ribulose 5-phosphate in presence of phosphoribose isomerase. This ribose 5-phosphate convert into ribose 1,5-diphosphate in presence of phosphoribonucleokinase. In presence of phosphopentose epimerase, xylulose 5-phosphate convert into ribulose 5-phosphate. This ribulose 5-phosphate convert into ribose 1,5-diphosphate in presence of phosphoribonucleokinase. Likewise, in dark reaction, CO2 is fixed to carbohydrates and the CO2 acceptor ribulose 1,5-diphosphate is regenerated. In Calvin cycle, 12 NADPH2 and 18 ATPs are required to fix 6 CO2 molecules into one hexose sugar molecule.